Hello student, this is Sarwar AD here. This is our third lecture on this chapter, the fishes, vertebrate success in water. Here it was a brief outline of the chapter. In these days we are working on. At the start we discuss about the evolutionary perspective in relation to phylogenetic relationship. Further, we slightly discuss about the survey of the fishes concerning to Agnathan and Gnathostom, and next we discuss about evolutionary pressure in relation to locomotion, nutrition, and digestive system yesterday. Today we will focus on the circulation and gaseous exchange mainly, and in the upcoming lecture we will discuss about the other thing mentioned in this outline. Here, when you say about the circulation in these organisms, they have closed circulatory system. Mean the blood usually flow in the vessel. It is not available or it is not moving in the open chambers, as like you have read about some invertebrate possess open circulatory system. Here these organisms possess closed circulatory system and a heart. The heart is very simple, but it's not so complex as it is present in the higher vertebrate or the mammals especially. Here, four embryological enlargement of the ventral aorta can easily be seen in the heart of the fishes: sinus venosus, ventricle, atrium, and conus arteriosus. These are the four enlargement of the ventral aorta, which are you can say designated different names, but they are present in a simple tubular form, so it's not so perfect heart as you assumed about the heart should be. Most fishes have single circuit circulatory system. Single circuit circulatory system and double circuit circulatory system, these are two type of the circuits in different organism having closed circuit circulatory system. Mean single circuit circulatory system, blood generally flow one time during the cycle to the heart, and if it is coming and going out once again coming in the heart and taking two cycle, the fish have single circuit circulatory system. And the lungfish, they also exhibit pulmonary circulation because they develop pneumatic sac or lung for their circulation along with the gill respiration or along with the gill gaseous exchange. And pulmonary and systematic circulations can easily be seen in these fishes. Here this is the diagrammatic representation of circulatory system of bony fishes and lungfishes. Here in the A section, these are different type of the arches, blood capillaries, and from the second to sixth, these are generally attached with the dorsal aorta and ventral aorta at some places in the bony fishes. तो इसमें जो है वो ये ऊपर वाला जो हिस्सा है ये बोनी फिशेस में दिखाया गया है, और जो नीचे जो बी वाली रिप्रेजेंटेशन है ये लंग फिशेस में है. लंग फिशेस में क्योंकि ये जो सिक्स्थ आर्च है ये जो है वो लंग्स को जो है वो पल्मोनरी आर्टरीज जो हैं ये लंग्स को जो है वो ब्लड सप्लाई करती हैं, जहाँ से मतलब ब्लड साफ़ होने के बाद वापस से दिल में आ जाता है. ठीक है जी. और इसमें अगर हम देखें तो यहाँ जो वेंट्रिकुलर सेप्टम है ये जो है वो सेप्टम जो है वो थोड़ा सा प्रोमिनेंट हो रहा है, लेकिन इन द बोनी फिशेस दिस सेप्टम इज़ नॉट सो प्रोमिनेंट और इवन नॉट प्रेजेंट. तो हम ये कह सकते हैं कि जो लंग फिशेस हैं इनमें जो सिस्टम है सर्कुलेटरी सिस्टम It is more developed as compared to bony fishes. Bony fishes generally respire through gill, but the lung fishes also respire through the gills, but occasionally they also utilize their lungs for gaseous exchange. Even though the lung fishes are not a transitional group, they provide a good example of how lungs alter the circulatory pattern. You can easily see that if any organism is going to respire through the lungs instead of gills, it needs some modification in its circulatory system or the circulatory patterns. So if the system is very simple, like the bony fish's circulatory system, then they don't need to have this type of the adaptation or this type of the amendment in their circulatory system. Here circulation to the gill continue, but a vessel to the lung has developed as a branch of aortic arch. Aortic arch sixth is you can say developed towards the lung for the circulation instead of the gills. However, other 5 arches are supplying blood to the gills for gaseous exchange. Here this is a simple representation of lungfish circulation. Here a few arches are going towards the lung, but this is shown here that one of them leads to the lung, where also take place the gaseous exchange, and further oxygenated blood once again carried out towards the heart. Further it moved to other regions of the body. Next we need to discuss about the gaseous exchange, water movement over the gill. We know that approximately the air contain more oxygen as compared to water bodies, and fishes live in an environment that contain less than 2.5% of the oxygen present in air. To maintain adequate level of oxygen in their bloodstream, fishes must pass large quantity of the water across their gill surfaces and extract a small amount of oxygen present in the water. It means they need to pass water over their gills for gaseous exchange to take place. Opercular and pharyngeal muscles pump water in most fishes. Opercular and the pharyngeal muscles pump water in most fishes. Ram ventilation in elasmobranchs and open ocean bony fishes. There are few fishes which use ram ventilation. Ram ventilation is a type of ventilation. Fish generally move by keeping their mouth open, and as they keep their mouth open, water flow through their mouth and exit through the gill chamber, and you can see gaseous exchange take place over there. This is the ram ventilation, and it generally occur in elasmobranchs. Elasmobranch belongs to the chondrichthyes. Gaseous exchange surfaces, we are well aware that visceral arches support the gills, and gills are the major sites for gaseous exchange in the fishes. However, gill filament and the pharyngeal lamellae, these are the you can say minute structure of the gills. Pharyngeal lamellae, these are the structures where there are the configuration of different type of the blood vessels, and gaseous exchange generally take place over there. And there are two different type of the mechanism. By employing these mechanism, gaseous exchange take place. First is the counter current exchange mechanism. There is the parallel current exchange mechanism. Here in this diagram, direction of the water is shown, and as you can say fish move in the water by keeping its mouth open, water enter inside the mouth and exit through the gill operculum or gill cover. Here closing or the operculum generally provides some pressure, and water move inside the chambers and inside the minute cavities of gill and lamellae, and water flow between these lamellae generally cause the gaseous exchange to take place by using counter current exchange mechanism or water flow. It means water and blood they flow opposite in directions. They flow opposite in direction. Here you can see the blood flow in this direction and water flow in this direction, and this is the contour current exchange flow mechanism. Most fishes have a muscular pumping mechanism to move the water into the mouth and pharynx over the gill and out of the fishes through gills opening. Muscles surrounding the pharynx and the opercular cavity, which is between the gills and the operculum, pump this power this pump. It means here where gills are present, different cavity of the opercula, because opercula is a opening and closing. By the time when fish needs to open it, they may open it slightly, but they may also close for creating negative pressure over there. In the previous slides we have discussed about the negative pressure mechanism in the fish for eating up something. Here the gills or the opercula, gill cover or the operculum, they help to create a power for this water current to move through the gills with pressure, so gaseous exchange take place. This happen in bony fishes, but in elasmobranchs or some bony fishes, opercula is not present, we know that. And when opercula is not present, it means that power can't be provided. And when opercula is not present, the power cannot be generated for this type of the exchange. Then fish always need to keep on moving or swimming in the water for the survival, and this is the ram ventilation we have discussed earlier, and in the next slides once again coming. Here these are the lamellas in a very general view, and this exchange is the contour current and this is the parallel current. Both type of the exchanges take place in water bodies. Here afferent and efferent blood vessel are shown. Afferent and efferent, one of them provide the muscle with blood and one of them carry the muscles away from the gills. Gaseous exchange of the pharyngeal lamellas. In the figure A shown the gill arches, and B, these are the trout lamellas are shown here, and in the C comparison of the contour and the parallel current exchange. This is the ram ventilation. In the ram ventilation, generally water flow through the mouth, and opercula are generally not present in these type of the fishes which generally utilize ram ventilation. And when they keep their mouth open and their cavity or the opercular cavity is always kept in a open position, and water just flow through the mouth and exit through the chamber where there should be operculum in the bony fishes, but in the elasmobranchs and other few fishes opercula are not present, hence water generally move out from these surfaces, and you can say gaseous exchange take place. Some elasmobranchs and open ocean bony fishes such as the tuna maintain water flow by holding their mouth open while swimming. This method is called ram ventilation. Here next, in few fishes, when you say that they don't have opercula for generating a power for gaseous exchange to take place by using the water, they possess spiracles. Just behind their eyes they possess spiracles. Here these are the spiracles shown just behind the eyes. There is a slight opening. These may be linked somewhere to the pharyngeal pouches or pharyngeal slits. Here spiracles are modified pharyngeal slits that open just behind the eyes of elasmobranchs and are used as an alternative route of water entering the pharynx. It means they are utilized in some fishes only for water entry. Here these are the pharyngeal lamellas. Lamellas generally contain blood vessels. Gaseous exchange occur as blood and water move in opposite directions or either side of the lamellas epithelial membranes. This counter-current exchange mechanism provide very efficient gaseous exchange by maintaining a concentration gradient between the blood and the water over the entire length of the capillary wall. Both type of the mechanism are shown once again. Next, we need to discuss about swim bladders and lungs. Swim bladders are the, you can say, extension. Pneumatic sac connect to the digestive tract in non-teleosed fish and function as lung in lungfishes, climbing perch and ancient rapidaceans. Climbing perch are the fishes generally may cross 1 pound in search of water to the other pond. Climbing perch are the type of the fishes you may search videos on the YouTube. You will see that this fish is in a situations to specifically Indian climbing perch. They generally cross 1 pound the other and they develop pneumatic sac which is very helpful for gaseous exchange while they are living on the earth. Function as swim bladders in other bony fishes. This is the Indian climbing perch shown in the lower diagram. Indian climbing fish spend most of its life entirely on the land, and these fishes, like most bony fishes, have gas chamber that is called pneumatic sac. So these structure help these fishes to be out of water for long time, moving in search of water. Most zoologists believe that the lungs are more primitive than swim bladder. Definitely, most of the early evolution of the bony fishes occurred in warm freshwater takes and streams during the Devonian periods. These bodies of the water frequently became stagnant and periodically dried. Sometimes the water bodies dried in harsh season. Having lungs in these habitat could have mean the difference between life and death. It mean if any organism or the fish living in such type of pond which are dried periodically and the fish which have adopted to perform gaseous exchange by using lungs or lungs type structure like pneumatic sac, they may sustain these conditions, but if they don't have, they surely will die over there, and hence it means this is the difference between life and death. On the other hand, later evolution of many modern fishes occurred in marine and freshwater environment where stagnation was not a problem. Definitely, when any animal is living in a large water body that is not seasonal like oceans, there is no any problem of being dried up periodically. In these environment, the use of the pneumatic sac in buoyancy regulation would have been adopted. Definitely, these fishes use these structure for moving or getting help in the buoyancy during the locomotion in water bodies. Here this is the diagram explaining the evolution of pneumatic sac and swim bladder. Possible sequence in the evolution of the pneumatic sac or the lungs, primitive lungs, just develop as a ventral outgrowth of the esophagus, and further some of them got disconnected from the main esophagus, or some of them get connected to the digestive tract or the gut tract, but some of them just remain attached to the main esophagus as a outgrowth, and but they just shifted to other functions. Swim bladder is generally filled with the gases while it is assisting in the locomotion by the gas gland.